Hello, everyone. Thank you very much for attending the virtual workshop entitled Metabolism-based Therapies for Epilepsy. My name is John Rowe. I'm a professor of neurosciences and pediatrics at the University of California, San Diego. And I'm going to be giving you a very brief overview of the topic of biomarkers relative to ketogenic diet action. So these are my disclosures on the Scientific Advisory Board activity, consultant activity, and let's first start off with a definition of biomarkers. We, in both the clinical and the scientific arena, use the term very loosely, and because of some inconsistencies in our interpretation, as well as use of these for both clinical and research purposes, a panel was actually convened by the FDA and NIH a few years ago to try to come up with a uniform consensus with regard to definitions and applications. So briefly defined, a biomarker is a characteristic that is measured as an indicator of normal biological processes, pathogenic processes, or responses to an exposure or intervention. Now, this is very deceptively simple and is much more complicated than that when it comes to actual applications. The types of biomarkers that are described in the document are listed here: diagnostic, monitoring, pharmacodynamic response, predictive versus prognostic, surrogate endpoints, safety biomarkers, and those that indicate susceptibility and risk. While any particular measure could encompass one or more of these domains, it's oftentimes important to actually validate this, and this is the framework that's been proposed for actually discovery development for so-called biomarkers of disease. Now, relative to the ketogenic diet, the key questions that have been outstanding for many decades are the following. What are the key features exhibited by patients who respond to the ketogenic diet? When the ketogenic diet fails in patients with epilepsy, do we know why? And this is above and beyond the issue of compliance. If patients are taking the ketogenic diet and they do not respond, do we know why? And then, are there reliable indicators of susceptibility to adverse side effects? Something as clinicians that we're all very interested in pursuing, and also very important to know. There haven't been very many studies regarding the whole concept notion of biomarkers. This is one of the only ones out there in the medical literature that looks at potential biochemical parameters and their association with response to ketogenic diet therapies. These investigators in the UK looked at various markers such as beta-hydroxybutyrate, acetoacetate, fatty acids, acetylcarnitine, and free carnitine profiles, as well as the glucose and glucose ketone levels. What they found was that the acetylcarnitine level was a significant predictor of response. And you can see here in the table, I'm looking at the p-values on the right-hand side, that several of them actually did show some statistically significant differences, ketone levels as well, in addition to the carnitine. And this suggests that metabolism is in fact something that can be addressed as a potential target for identifying biomarkers. So the first question, which has been around for nearly as long as the ketogenic diet has been with us, the concept notion, as the name implies, ketone bodies. Do ketone body levels correlate with seizure control? The data here, both experimentally and clinically, are mixed. We don't have a definitive answer to this. Some recent data suggests that perhaps there is an inverse correlation with seizure frequency as well as ketone levels measured in the blood. But the problem here is that, we oftentimes in clinical practice measure urine ketones, which are basically measuring acetoacetate, blood ketone levels, which do not necessarily reflect brain levels or compartmental levels. And then there's been some research on breath acetone as an indicator of systemic ketosis. This is an area that still requires a lot of investigation to determine whether, in fact, ketones are actually related to ketogenic diet clinical effects. What about genetic predictors of efficacy? The only real study out there that's looked at this in some detail is this paper that was published by, again, the UK group in collaboration with Mass General and other centers in the US. They looked at the variants of two genes, KCNJ11, which encodes the potassium channel that's modulated by ATP and ADP levels, and BAD, which is the BCL2 agonist of death, which is a proapoptotic factor, but also linked to glycolysis. They didn't find anything that really predicted response to ketogenic diet therapies, despite the notion that the BAD regulation and actually has a potent effect in animal models of seizures and epilepsy. Can we learn something from failures? So when the ketogenic diet fails in patients with epilepsy, do we know the reasons why? We often think of compliance, obviously, but is there a relationship with age, sex, skill level of the individual team that's actually administering the ketogenic diet, or the various formulations that are utilized? What are the underlying neurobiological reasons? Could resistance reflect the state of disease ontogeny, especially with respect to ongoing or progressive metabolic derangements? And I think that at this point, we really don't have any reliable direct biomarkers or even surrogate markers that give us a clue in terms of why there's failure. Can we learn something from anti-seizure drug failures? In other words, the lessons from pharmacoresistance. Over the years, there have been a number of hypotheses that have been advanced, including receptor desensitization, turnover, downregulation, drug efflux mechanisms, such as through P-glycoproteins. And there's even a metabolic hypothesis that suggests that the intrinsic metabolic state of the brain may actually predispose not just to efficacy, but also to failure. And along these lines, do clinical disorders where we treat patients with the ketogenic diet, where they're contraindicated, for example, do they teach us anything about this? Are there epigenetic or genetic factors underlying these disorders that perhaps can give us some clues? Many of you know that the ketogenic diet is contraindicated in fatty acid oxidation disorders, carnitine transport disorders, and a couple other rare metabolic conditions. There are relative contraindications based upon sort of the psychosocial, operational, and logistical constraints of actually administering a diet, which have been summarized in this consensus statement published a couple years ago. But going back to sort of less learning from successes based upon the target population that we use, what insights can be derived from special populations that appear to respond favorably to the ketogenic diet? And here, clinical experience has taught us that there are a number of disorders that appear to have a fairly good response to the ketogenic diet, defined by either 50% or greater seizure reduction. But if you look at the list here, you can see, for those of you who understand some of the biology and the clinical attributes of these disorders, they're very different in many respects. And so one of the hypotheses that's come out is, does the ketogenic diet work through some common final mechanism to reduce seizures? And the relative indications, this is the list of conditions that have a better than average dietary response with less clinical evidence of efficacy. But you can also appreciate here that the heterogeneity of these conditions is also markedly large. The question that's been asked over the years, can we predict a favorable response to ketogenic diet therapies for drug-resistant epilepsy? Well, the answer is no. There are as yet no strong studies or evidence that suggest that specific factors can affect response or even tolerability issues. And why is that? Well, we're looking at not a drug therapy on a molecular target that seems to have some downstream effects within the cells, or even within the tripartite neuron astrocyte microvascular compartment. Metabolic pathways are intrinsically very complicated. And while notable attention has been given to glycolysis, fatty acid oxidation, pentose phosphate shunt, tricarboxylic acid cycle, as well as oxidative phosphorylation in the mitochondria, many of these interactions are bidirectional and very complex with regard to dissecting out what is actually direct causally related changes as opposed to epiphenomena or systemic metabolic network changes. And then we have to remind ourselves that epilepsy is a moving target, that while there may be susceptibility and exogenous injury that either alone or together combine to produce the cascade of molecular, cellular, and perhaps genetic and epigenetic changes that lead to the chronic state of epilepsy, these take time, basically over minutes, days, weeks, and months, perhaps years in some cases, particularly for post-traumatic epilepsy. So in a great sense, this epilepsy is a moving target. And when applied to the developing brain, it's even more complicated because we're juxtaposing normal brain development with pathological processes and some of the effects that we see. And of course, many of you are familiar with the age-dependent epileptic syndromes that we see in the pediatric population that again challenges us in terms of determining how and when these things occur and what the factors are that influence the clinical state. Many epilepsy syndromes have been identified and studied over the years. And of course, the challenge here is to identify one or more where we get true insights of ketogenic diet therapy in terms of developing biomarkers. So in summary, we asked the question, what are the key features exhibited by patients who do respond to the ketogenic diet? Well, there is no consistent phenotype or set of clinical or experimental biomarkers or surrogate markers at the present time. When the ketogenic diet fails in patients with epilepsy, do we know why? No, not really, but this is something very important to pursue. And are there reliable indicators of susceptibility to adverse side effects other than contraindicated disorders or the instance of noncompliance? There are none. So thank you for your attention. We look forward to engaging further in our dialogue on this very important topic.